everyone, and welcome to Research Recap, brought to you by Arbor Assays. In this series, we dive into fascinating studies conducted by scientists across the globe, exploring how they're using Arbor Assays kits to unlock new discoveries. I'm Cassie Shoemaker, Sales and Marketing Manager here at Arbor Assays, and I'm excited to highlight the amazing work being done in labs like yours, share insights, and take a closer look at the tools driving innovation and research. So let's get started. Today, I'm joined by Dr. Emily Elwell. Emily is a researcher from the University of Wolverhampton in the UK, who studies the chemical communication of lemurs and how this can be applied to conservation breeding in zoos. Welcome, Emily.
Hi, thanks for having me.
Thank you so much for joining us. Can't wait to hear more about the studies that you got to do with the lemurs. Can you tell us a little bit about some of the research questions that you were hoping to find and answer with these studies?
Yeah, the important thing, sort of background to know about my research, is that lemurs communicate extensively with scents and smells, which we call chemical communication. And we know this chemical communication basically can signal things and convey information about lemurs. So it can give information about sex, dominance, rank, or social status. Crucially, it can signal fertility in females and give cues of when the breeding season is. You can actually collect these scents because lemurs scent mark everywhere, kind of like how a cat rubs things if you have one in your house. And you can analyze this with gas chromatography, mass spectrometry, and you can actually identify individual chemical compounds. This genus lemur has two distinct species in it. They're made up of the red ruff lemur and the black and white ruff lemur. They are closely related, and originally I got the scents I collected were from red ruff lemurs, but we wanted to extend the study to black and white ruff lemurs because we believe that their communication is similar. They show the same scent marking behaviours, so we think there should be overlap. And historically in Madagascar, their home ranges did overlap, and there is some evidence for hybridization. So we think that there should be some overlap in the chemical communication. My main research questions was, are there actually any specific chemical compounds in female ruff lemur scent marks that actually signal fertility or provide a cue to the breeding season? And we asked whether or not we could actually just create this in the lab as a form of enrichment for zoo lemurs, and then if we present it, we actually trigger a mating response in male lemurs.
Yeah, that's wonderful. And has there traditionally been issues getting the lemurs to reproduce in captivity?
So lemurs are one of the most endangered groups of animals in the world. Both the species of ruff lemur that I've studied are critically endangered and are considered to be conservation priorities. Zoos kind of help conservation by basically providing almost a buffer for extinction and a pool of genetic diversity for future reintroduction. So actually breeding within zoos is really important. But a lot of species don't necessarily breed as well as they do in the wild, and ruff lemurs are one of those species. So it is important if we can think of ways to actually improve breeding programs and kind of secure the future of the species.
That's really important. We want to make sure we take care of such a precious species. So can you tell us a little bit more about the biomarkers that you were also looking at? So you were looking at the composition of the scent, but you were also looking at some other biomarkers. Yes, can you tell us a little bit about what you chose?
So the biomarkers are mainly looked at actually after we produce the scent. So we did manage to collect the scent and identify sort of what we think is a cue of the breeding season. So we collected scents in the breeding season and the non-breeding season, compared them because lemurs do have kind of a set window when they breed. So this does make that a lot easier because it is a bit more predictable. So we actually presented the scent in zoos that we made. We identified 12 chemicals that are commercially available to buy because they're just found on plants, basically. Mix them together in a kind of a mixture a bit similar to almost like essential oils, which is what is kind of used in zoos as a sort of scent enrichment rather than a sort of biological one. We presented this to different groups of rough lemur. Just basically, we just dipped cotton strips in the scent mixture and hung it around. So as I said, crucially looking for if it could actually trigger male mating behaviours outside of the breeding season. So we actually did this in the summer. But on this as well, we also collected fecal samples and we did test males for testosterone. There's been a few studies in the wild and in zoos of other species of lemur and other small primates, and they have found that in the breeding season, male testosterone concentration does increase. It's quite significantly. So we kind of thought if our kind of mixture is actually sort of recreating this cue that then should sort of create these similar breeding season conditions. So we would expect the testosterone to go up in males as well.
Great. Yeah, that's wonderful. So I know you were just describing how you were looking at the male feces for an increased level in testosterone, and you used the Arbor Assays testosterone kits to do that. Can you tell us a little bit about your introduction to Arbor Assays as a brand, or Arbor Assays, these particular ELISA kits, why you selected them, and what may have been helpful to you with them?
Yeah, so actually it was introduced to me by a colleague who was a postdoc, and this was from my PhD work. So at the time, in my postdoc, and she trained me to use assay kits, and she was testing fertility in female lemur. So she was using a progesterone kit from yourself. So I was actually trained with the Arbor assay kit, and she had done previous work that had been published, and she used these same kits, obviously, because we're in the same lab as well. We wanted to keep it consistent and use the same kits, but from knowing her experience, I knew they would be reliable. Had tried some other commercial kits as well, and that she also trained me on for cortisol, I think it was, but I had to say I found that the Arbor Assays one I much preferred. I found it a lot easier to use. So when it came to sort of actually doing my full kind of study, I decided to go with yourselves.
Wonderful. I'm glad to hear that you found our kits easy to use and that you heard about them from someone else in your lab. Okay, wonderful. So you found the chemical mixture that you wanted to try, and you had your research questions. So tell us a little bit about what you found.
Okay, so like I said, we found 12 sort of compounds that we think was a key to the breeding season. We used those to make an enrichment. We had 3 periods, so a baseline period when I took behavioural observations and collected fecal samples for analysis for testosterone, and that just kind of set what was normal for those groups and those individuals. We then had the experimental period where we, as I said, dipped cotton in the scent mixture and placed it around. And then following that, we had like we call it a post-enrichment period, which is typical in these kinds of studies, and it was just to see if any of the effects just actually continued once the actual cue was no longer there. We did find that mating was triggered in all the groups that we studied, and it was triggered in all males except for the oldest male. So this is really positive. As I said, this was outside of breeding season. We wouldn't usually see these behaviours. We found all types of sexual behaviours increase. So this was sort of mounting of other individuals, almost display behaviours that the males kind of do towards females in the wild. We saw a little bit of those, which was quite interesting and strange as well. And we also found males sniffed female genitals more frequently once the scent was there. We did also find an increase in testosterone, but this didn't actually occur until the post-enrichment period, so after the sort of exposure to the scent. It wasn't significant, but it was a clear increase, so we think it was just a matter of the sample size, which is a little bit annoying. But we found the pattern in the majority of males, so there was a huge increase. So I think there was a delay after the scent, a bit of a physiological delay before it kind of shown. So it would have been interesting to continue to collect because we think it would have definitely made a difference. Surprisingly as well, we did actually test, they sent me the all bachelor male group. And this is because we did assess it for welfare effects as well while we were there. So it was more for the welfare, but it did actually trigger mating within a male group, and surprisingly, it was actually had the highest number of mountings in the male group compared to the actual breeding groups, which was quite interesting.
Yeah, that is interesting. Maybe a little unexpected, that particular finding. But that's wonderful that your compound did seem to be able to, within those breeding pairs that maybe had trouble in captivity, otherwise wanting to breed, that it did seem to make a difference.
We did actually result into unexpected pregnancies as well, following the study. So that was quite exciting.
That's
there were
really
also
exciting.
quite a few dominance fights between males in the breeding season following as well, which obviously we don't know for sure, but the zoos felt that it was related because it was unusual.
Yeah, yeah, that's great. So thinking back over all of the results that you found in this study in general, do you have three kind of main messages or takeaways that you would like to share with anyone watching for if they wanted to reproduce, or just other takeaways that they had as they look to recreate your study or add on to it?
So my first would be to collect more samples, just collect more samples than you actually need, because what you think you need is probably not enough. Or there are things I lost some samples along the way when I was processing them and things, just because of the nature of the animals. So I would say always collect more than you actually think you need, because it's better to have more, because you never know if they're no good until you actually come to analyze them. Figure out what you want to find out first and then move backwards from that. So you need to know what you need to collect before you just sort of start doing it. Because you often think in hindsight, once you started it, you start reading, someone else did this, and you think, oh, maybe that could have been better. Like when I collected the scents, there were some issues with the pandemic, but in hindsight, I wish I'd also collected female fecal samples and tested those for progesterone as well, just because it would just make it more clear to just illustrate when the female was actually fertile, rather than just saying this was in the breeding season.
Yeah.
And for people studying in my field, studying behaviour, I've noticed a lot of papers and research, it's just purely behavioural, but I would say to try and avoid that. If you want to do something in this field, you should really try and combine behavioural measures with physiological indicators, whether that be hormone testing or things like I've seen some papers that test heart rate and things, because kind of just animals can hide things, and it's very easy just kind of project sort of human behaviors on animals, so it's very subjective. So I just think if combining it with something physiological just gives you so much more information and just better accurate information as well.
Yeah, absolutely. Being able to make a little bit more quantitative judgment on an otherwise qualitative assessment. Yeah. Thank you so much for tuning in to this episode of Research Recap. We hope you enjoyed exploring the exciting research that was done by Emily and her colleagues. If you have any questions for Emily, please feel free to contact her at the information shown on the screen. If you or someone in your lab has published work using our kits and would like to be featured in a future episode, please be sure to email us at marketing@arborassays.com. Until next time, have a great day, and we'll see you in the next Research Recap.